The biotech firm Moderna Therapeutics announced this morning that the first 8 participants in the first phase of its COVID-19 vaccine trial developed some antibodies after just one single dose. Now, that's a promising sign from the trial done in collaboration with the National Institutes of Health. Moderna was the first U.S. company to test its vaccine on humans. That was back in March. It's expected to start a second round of clinical trials as soon as next month. First, on CBS This Morning, the president of Moderna, that's Dr. Stephen Hoge, joins us to discuss. Dr. Hoge, thank you. It's really good to see you. You know, they've got the best and the smartest minds in the world working on this vaccine, and word is that many of them work at your place. So could you explain exactly what you all discovered in the first trial?
Sure. Well, Gail, thank you for having us. So this morning we were very pleased to announce the positive interim phase 1 data, which showed that all subjects who had received 2 doses of our vaccine in the first 2 dose level cohorts, that's at 25 microgram, a low dose, and 100 microgram, our mid dose, had seroconverted to develop antibodies in their blood that bound the virus. And importantly, actually, when we characterized 8 of those subjects and said, would it neutralize the virus in a viral replication assay, it showed that those same antibodies that those people had developed actually could neutralize the virus and prevent its ability to infect human cells. Altogether, we're very pleased by that result because it suggests we're on the right path with this vaccine.
Now, you said you started with 8 participants. How many participants do you need to show, we have something here? 8 seems like a small number. Is that pretty normal?
So 8 was just a neutralization assay test. Actually, we've started with our colleagues at the NIH who are running this phase 1 study, 45 subjects across 3 dose levels. So there's 15 at the low dose, the mid dose, and the high dose. And what we announced today was actually we have binding antibody data from the first 2 cohorts, each of 15 people. So it's a total of 30 people already. And so we're very pleased to see that all of those people had seroconverted. And actually, they've seroconverted to levels of antibody in their blood that are at or above the levels that somebody who's recovered from the virus, it's called convalescent sera, the blood of somebody who's recovered from the virus have. So we've already exceeded what it takes, at least by that measure. Now, the 8 that you referenced before, Gail, those are folks where we went to that extra level and said, can we characterize the quality of that antibody response and show that it actually would have been protective against infection in human cells? And in fact, all 8 out of 8 of those did. So we're quite pleased with the breadth of the immune response so far.
Okay, I'm not a doctor, but that does sound very encouraging. So what happens now in phase 2?
So in phase 2, our responsibility is to expand the safety and immunogenicity database, and it's really a primary focus on safety. The phase 1, the vaccine was generally safe and well tolerated, but 45 people is still a relatively small number. And so we'll be looking across 600 people in the phase 2 study, which will start any day now. And that will be to both confirm that we've got the right idea of the dose, where we believe we're going to see protective immunity, as well as that the safety profile continues to hold up.
There still seems to be so much, Dr. Hoge, that we don't know about this virus. So how do you develop an efficient vaccine when it doesn't seem like we're quite sure what we're dealing with yet?
Yeah. Well, one of the great things that we do know is we can study what's happening to people who are recovering from the virus. Where are the antibodies and immune system getting a hold of the virus to essentially clear the disease? And we have learned from what's been published to date in terms of infections is that generally people aren't getting reinfected. When they recover, their immune systems have been able to handle that virus. When you look at that particular question, what we discover is that there are proteins on the surface of the virus, particularly one called the spike protein that we've all learned a lot about. And that spike protein, antibodies to it appear to be protective in many people who've recovered from the virus. And so the approach we've taken at Moderna and other vaccine innovators and manufacturers have taken is to try and express that spike protein so that the immune system can learn to recognize it and protect against the virus. Now that approach we think has a high probability of success in the vaccine context, but it still doesn't capture all of the complexity of the disease, to your point, that we see. In fact, as we see the virus in natural infection in humans, we're seeing all sorts of surprising things that the virus can do. The good news is, though, that despite all that diversity of things, we do believe this approach of going after the spike protein will actually address all of those diseases and hopefully prevent many of those findings.
Okay, we all want the vaccine, but we also want it safe. What's your best guess on when we'll have something on the market quickly? What are you thinking? Your timeline?
Well, we're starting the phase 2 imminently. As we said today, we're starting the phase 3 by early summer, which is just a couple of months away. And our goal is to have a vaccine available for broad distribution by year-end or early next year. Now, the question, of course, that's out there is, as we start to build data over time, and if we and others build data that shows that the vaccine has a potential for benefit, that it's safe and has a potential for efficacy, then of course there are circumstances where the vaccine could be deployed to high-risk populations earlier under something called an emergency use authorization. Really, though, that's a decision that regulators, in particular the FDA, have to make.
All right, but I'm focusing on, you said year and next year. Thank you, Dr. Stephen Hoge, and good luck.